Hello, so today we'll be talking about hormonal control of energy metabolism. So this is a part of the lecture series on physiology of everyday life. Control of energy metabolism, this is the introduction slides. We'll be coming back to this again, so don't be worried about it. So it is a busy slide, you can see that, but what happens is there is glycolysis, there is gluconeogenesis and glycogen metabolism, fructose metabolism, lipid metabolism, ketone and protein metabolism. All these metabolism are integrated with each other, and for that integration there must be something which is controlling the inflow and outflow of intermediates from one pathway to another. So we'll be coming to that. Glycolysis is the breakdown of glucose to pyruvate. Breakdown of one glucose to 2 pyruvate is known as glycolysis, that is converted to acetyl CoA, which enters the TCA cycle, which is the tricyclic acid cycle. Gluconeogenesis, what is gluconeogenesis? Gluconeogenesis is the formation of glucose molecules from non-carbohydrate substrates. So one thing I want to point out is that acetyl coenzyme A will never form glucose, so it will never be gluconeogenic. In proteins, amino acids, 18 out of the 20 amino acids are gluconeogenic. मतलब कि they are either purely gluconeogenic or gluconeogenic plus ketogenic, but 18 out of 20 are gluconeogenic, so only 2 are there which will not form glucose. All of this we'll be talking about in details. Okay? So there is glycogen metabolism, like glycogen synthesis, glycogen breakdown, fructose is entering into the glycolytic pathway, there is lipid formation, there is lipid breakdown, and there is formation of ketone bodies. So how does this control happen? There is one major crude level of control which is taking place, which is known as compartmentalization. So in compartmentalization, what happens is, in cytoplasm there is anabolic pathways which are happening. We'll talk about anabolic pathways. And in mitochondria there are catabolic pathways which are happening. Two exceptions are the gluconeogenic pathway, which is a catabolic pathway, is happening in cytoplasm, and glycogen breakdown, which is a catabolic pathway, is also happening in the cytoplasm. So these are the two exceptions of catabolic pathways which are happening in the cytoplasm. Otherwise, mitochondria is the place where the catabolic pathways takes place. This is the first crude level of control which is taking place, which is compartmentalization. Then there are 3 more level of controls, which is substrate level of control, allosteric modification and covalent modification. So substrate level of control is a very primitive level of control and very simple to understand. Like if this glucose 6-phosphate to fructose 6-phosphate, it is a reversible pathway. So if glucose 6-phosphate is more, it will favor the conversion of glucose to fructose. And if fructose 6-phosphate is more, it will favor the conversion of fructose to glucose. So that is how this reaction is controlled. In allosteric modulation, what happens is the enzyme has a site for the allosteric modifier, which is different from the site which is different from the substrate binding site. So what happens is that the allosteric modifier binds to the enzyme, and that enzyme is modified, either activated or inhibited. The covalent modification, what happens is the covalent modifier modifies the enzyme, either phosphorylates or dephosphorylates in most examples, and it either activates or deactivates the enzyme via this covalent modification. So there are 4 levels of control which we have talked about. One is the compartmentalization, second is substrate level, then allosteric modulation, and the covalent modification that happens. Okay, so this was the overview of what we'll be talking about. We'll come back to this in quite detail. Okay, so what is metabolism and energy balance? What is that we'll be talking about in quite detail is, metabolism is nothing but all the reactions which are taking place in the body is metabolism. So energy balance is the first law of thermodynamics, which states that energy can neither be created nor be destroyed. It can only be converted from one form to another. So that is what energy balance is. It's defined as the state achieved when the energy intake equals the energy expenditure. So in metabolism, it is the highly integrated network of reactions that provide cells with metabolic energy, which is the ATP or the creatine phosphate. Reducing power, which is the NADH or the FADH molecules, which will go into the electron transport chain and produce the energy, which is the ATP or the GTP or the creatine phosphate. And biosynthetic intermediates are the intermediates which are required. For example, acetyl coenzyme. Acetyl coenzyme is required to form the ketone bodies or required for triglyceride synthesis. So these biosynthetic intermediates are also found in as a part of the integrated network of reactions. So in this lecture will not be discussing about the intricacies of all the reactions by the biochemistry point of view. We'll be talking about more as in the physiological point of view and how these reactions are integrated with each other and what is the physiological basis of all the reactions which are taking place. So what is anabolism and catabolism? Anabolism is the synthesis of larger molecules from the smaller precursors. Catabolism is the breakdown of larger precursors and larger molecules into smaller ones, mainly involving the oxidative reactions. And there is also one thing known as amphibolic pathways. Amphibolic pathways are in which anabolism and catabolism both takes place. So that is also like gluconeogenesis can also be taken as amphibolic pathway. First there is catabolism and then there is anabolism of glucose. For example, here we can see like if the protein metabolism, the amino acids are catabolized first, then they go to alpha ketoglutarate. Alpha ketoglutarate forms acetyl coenzyme, not acetyl coenzyme, oxaloacetate. That oxaloacetate can go into phosphoenol pyruvate and that goes to the glucose via the gluconeogenic pathway. So first there is catabolism and then there is anabolism of glucose. So that is also an example of amphibolic path. So what actually is energy balance? What energy balance we talked about from the physiological point of view, it is just the energy input is going into the output and there is something which is stored. So energy input goes into output and storage, that is the energy balance. If input is more than the output, then the excess energy will be stored, that will lead to weight gain and something like that. Okay. So carbohydrate metabolism, carbohydrate acts as the energy input, protein acts as the input and fat. So these are the three major macronutrients which act as the energy input. This is the last time we are talking about them as the inputs. Otherwise we'll be talking about them only as glucose. Carbohydrate will be glucose, protein will be amino acid and fats will be fatty acids or triglycerides. Okay, because we are not talking about digestion or absorption, we are just talking about the metabolism after the absorption has taken place. So what is energy output? Energy output is in the form of mechanical work, which is the physical work. This is the voluntary work which we can increase or decrease. So 10 to 20% of the total energy output is in the form of voluntary skeletal muscle activity, which we can control. So if we want to lose weight, then this is the one which we can increase or decrease by our will. Then there are synthetic reactions which we talked about, biosynthetic intermediates. There is membrane transport which requires a lot of energy. There is signal generation and conduction. So signal generation and conduction via the action potential which is generated, and then there is sodium potassium ATPase which is required to reform the resting membrane potential. That is a very energy consuming process. So signal generation and conduction, the brain almost takes up 20 to 30% of the basal metabolic rate, just the brain, because there is a lot of signal generation and conduction happening there. Then there is detoxification and degradation. So liver also takes up 20 to 30% of the basal metabolic rate. So all of this is basal metabolic rate. And then there is heat production. So heat production is in a darker color because heat production can be voluntary and there can be involuntary heat production, which is when there is energy which is being converted from one form to another. There is involuntary release of heat. So that is what the heat production is involuntary. But there is voluntary heat production which is in the form of thermoregulation. When there is cold and warm-blooded animals need to maintain a body temperature, so that is when the heat production at voluntary level also takes place. Okay? So that is what the energy input goes to output and excess is stored. Now we'll be talking about fed versus fasting state. So I have chosen as fed and fasting because this is more conventional terminology, but the better terminology to be used would be fed is better to be termed as absorptive state, fed state as absorptive state, and fasting state to be considered as post-absorptive state. So fed is absorptive state, fasting is post-absorptive state. So in fed state what happens is there are nutrients. So these nutrients are converted into energy. So there is utilization of nutrients for energy, and the excess nutrients are stored in the body stores. So what I want to tell you about now is from evolutionary perspective, hunter gatherers, they didn't have energy or food excess 24/7 throughout the day, but the cells of the body and all the organs of the body require energy 24/7, but the energy was not available. So what happened was the nutrients was consumed, and when the nutrient was consumed, the small amount of nutrients was used for energy, and rest excess was stored as energy, so that when there is fasting state in which no nutrients were available throughout the day, then the body stores can be utilized for energy consumption. Okay, so that is, and today also, not even hunter gatherers, today also, even though there is surplus of food available in most parts of the world, the socio-occupational reasons, because of the socio-occupational reasons, we don't eat throughout the day. So this is valid now also, that the fed state, in the fed state we consume energy, and then we stored in the body stores, and in the fasting state what happens is these body stores are used for energy utilization. Okay? So this is the crux of what is happening in what we'll be discussing about in today's lecture is fed state, energy is utilized and excess is stored. In fasting state, the stores are used for energy. Simple. So what are the forms in which these body stores are stored? Necessarily, these are three important types. One is the glycogen, second is protein, and third is lipid. Okay? So protein is not necessarily a form of energy store. Protein is used for much more functions, except it is used for much more functions over and above the energy storage. So mainly there are two, glycogen and lipid. In a 70 kg adult man, around 1% of the body weight can be stored as glycogen. So 700 grams can be stored as glycogen, and energy density from the glycogen is not 4, which is from the carbohydrate, but is a 1 to 2 kilocalories per gram. Why is that? Is because, for example, take the example of powdered soup and tomato soup, which is canned soup. In canned soup, the water is already present. So when I want to store canned soup, I can store a small amount of energy, and we are talking in terms of energy. I can store a small amount of energy in a particular space, smaller amount of energy in a particular space as compared to powdered soup. So glycogen is necessarily the same thing. Glycogen is a hydrophilic substance, but lipid is a hydrophobic substance. So lipid is stored without water, but glycogen is stored with water. So this is not just the weight of energy density of the glycogen, this is energy density of the stored glycogen along with the water. So that decreases the effective energy density practically from 4 to 1.5 kilocalorie per gram. So the energy from the glycogen can only be 1000 kilocalories, and an adult man, 70 kgs adult man, requires about 2000 kilocalories per day. So glycogen alone can only sustain you for about half a day. But from lipid, what happens is lipid is around 20% is stored in the form of lipid. So weight 70 kg mein the lipid would be 14 kgs, and the energy density is the maximum 9.5 kilocalories per gram. So the energy stored in the lipid is 131,000 kilocalories. So if there is enough availability of water, the lipid can sustain you for about 30 to 60 days. Proteins are only used when there is nothing available. Proteins are about 10 to 15%, so which comes out to be 10 kgs, but half of the proteins would be for structural function and will never be used for energy consumption. So that the rest half, 5 kgs into 4 would be 21,000, can sustain you for about 10 days of starvation. So we talked about the energy stores, but one thing to understand is, if the lipid is so energy efficient, it has like 9 kilocalories per gram, it can sustain a lot of, and there is no limit for lipid storage. Why does the body even bother to store the energy in the form of glycogen? That is the question that arises. That is because some organs prefer some different fuels. So there are different fuels preferred by different organs in different states, like fed state, fasting state, and starvation. So to remember that easily, we'll consider in fed state everybody uses glucose because there is a plenty of glucose available in fed state. Everybody uses glucose except the heart. Heart is a hard working. Remember it like that. Heart is a hard working organ because it beats 72 times, 60 to 100 times per minute throughout our life. Requires a lot of energy, and fatty acid consumption is much more efficient than glucose consumption in production of energy. So heart uses fatty acids even in the fed state, rest all will use glucose. One thing is clear. Now second, in fasting state what happens is now the glucose availability is very less, but there is stored lipid, there is storage of lipid, a lot of lipid is being stored. So now the lipid is broken down into fatty acids, and everybody uses fatty acids for their energy consumption in fasting state except brain and RBCs. Now remember about the RBCs. RBCs don't have mitochondria, so they cannot oxidize anything. So the only thing that they can use as energy is glucose, because glucose can be used anaerobically in glycolysis. So glucose can be used anaerobically in glycolysis. So RBCs are helpless, so they can only use glucose. The brain is a high maintenance organ, so even when there is low availability of glucose, brain still uses glucose, and that is why the body was storing the glycogen, because of the glucose dependent organ. Some organs use glucose throughout the day, and brain also requires glucose. In starvation what happens is, remember two things, liver and muscle. Liver is a selfless organ. Liver produces ketone bodies, but it never consumes ketone bodies. It produces ketone bodies so that other organs can use ketone bodies. Muscle is a kind of selfish organ because muscle consumes everything: fatty acid, amino acid, ketone bodies. Anything and everything that comes by muscle, it uses it for energy. Glycogen, which is stored in the muscle, never contributes to blood glucose levels. That glycogen is only stored in the muscle and then used by the muscle for its energy when there is starvation or fasting state. Second thing, adipose tissue. Adipose tissue is only, throughout the lecture we'll be saying that adipose tissue is mostly concerned with only fatty acid. So it uses glucose when there is fatty acid when fasting, fatty acid when starvation, most of the time. So now we know that brain and RBCs, and especially brain, uses glucose throughout the day and even in the fasting state. We need to know how the level of glucose alters throughout the day. So sources of blood glucose during the day would be during the breakfast when we take the meal, fed state. 100% of the glucose comes from the diet. The x-axis there is time, which is the breakfast, lunch and dinner. On the y-axis there is source of blood glucose during a normal day. So when there is breakfast, the 100% of the glucose is coming from the diet, and when the time passes by, the dietary glucose decreases and the glycogenolysis increases. So the glucose is coming from glycogen stores, and then again the lunch happens. In the lunch we take the meal, so the source of blood glucose is from the diet, and then same thing happens, dietary glucose starts to decline and glycogen genolysis starts to increase. But after dinner what happens is, after the dinner 100% of the glucose came from the diet, but the glycogenolysis can only sustain mainly for 6 to 8 hours. So after 6 to 8 hours the glycogenolysis contribution to blood glucose will start to decrease, and the gluconeogenesis will also start. Overnight there is increased gluconeogenesis so as to maintain the blood glucose levels. Understood? Then we'll be coming now to the main crux of what the lecture is about: metabolism in fed state, fasting state and starvation. Okay? So this appears to be a very busy slide, but we'll take it one by one. So there are three organs which we'll be talking about mainly, which is the liver, muscle and the adipose tissue. So in fed state what happens is, like intestine gives three things to the body. In the blood, the three levels of glucose, amino acid and the fatty acids increase, and fatty acids increase in the form of chylomicrons, are absorbed in the form of chylomicrons. So what happens in the liver is, from intestine glucose comes and GLUT2 and GLUT4 are activated on the liver. Mainly the GLUT2 The GLUT2 glucose will be taken up. It is facilitated diffusion. GLUT2 glucose will be taken up by the liver, and the liver will convert that glucose take part in the glycolytic pathway, form pyruvate. Pyruvate will go to acetyl coenzyme A. Acetyl coenzyme A will go to TCA cycle. So glucose providing energy in the form of TCA cycle. One. Second, excess glucose is stored in the form of glycogen. So there is glycogen synthesis. Third thing, what happens is there is limited amount of glycogen which can be stored in the body. So excess glucose which is being utilized, and after the glycogen storage will go into lipogenesis. Lipogenesis is lipid formation. So acetyl coenzyme A will go into lipid formation, and that the fatty acids will be bound to glycerol 3-phosphate, which is either coming from the capillaries of the adipose tissue, we'll talk about that later, or the intermediate of the glycolysis. Or coming from the capillaries of the adipose tissue, the glycerol 3-phosphate will bind to the fatty acid and form triglyceride. These triglycerides will be packed in the form of VLDL and sent to adipose tissue for storage. So this is what is happening in the liver. What happens in the muscle is, glucose is taken up by GLUT4 transporter, which is insulin dependent. So glucose will be taken up by GLUT4 transporters, which is insulin dependent. Then the glucose will go into glycolysis, go into TCA cycle and provide energy to the muscle. Excess will be stored as glycogen. Also in the muscle, there is protein turnover taking place. What is protein turnover? Protein turnover is there is breakdown of protein happening, and also there is formation of protein happening. So for formation or synthesis of new proteins, the muscle will take up amino acids. So muscle is taking up amino acid and forming protein. So there is protein synthesis. Also under the effect of insulin, the potassium channels are also activated. The potassium channels activated, potassium will go inside. So this has a very important clinical relevance. Why? Because if the patient is in hyperkalemia is increased potassium in the blood. So one of the modalities by which we can treat hyperkalemia is give the patient insulin, because insulin will activate the potassium channels. The potassium will go inside the muscle cells and the potassium in the blood will decrease. But the insulin will also make the glucose go inside the muscle cells. So if we give insulin alone, the patient will be treated for hyperkalemia but might land up in hypoglycemia. So what we do is we give insulin along with glucose. So insulin plus dextrose is a treatment for hyperkalemia. You understand why? Now, in adipose tissue, what is happening is, this is an adipose tissue. In adipose tissue, not much will be happening. It will only be concerned with fatty acid. So chylomicron is coming. Now the chylomicron which comes to the capillaries of the adipose tissue, there is presence of lipoprotein lipase on the endothelium of the capillaries of the adipose tissue. So this lipoprotein lipase is activated by insulin. Lipoprotein lipase will break down the triglycerides in the chylomicrons into fatty acids and glycerol 6-phosphate. The fatty acid will enter into the adipose tissue via just diffusion, or there is transporters also, fatty acid transporters also present on the adipose tissue cells. So the fatty acid will come inside, and then the glycerol 3-phosphate, which was the intermediate of the glycolytic pathway, will bind to the fatty acid and stored as triglycerides. The glucose will enter the adipose cell via the GLUT4 transporters, which is also insulin dependent. Glucose will go into glycolysis, and then glycolysis will go into the TCA cycle to provide energy. Excess of acetyl coenzyme A will go into lipogenesis and form fatty acids and bind to glycerol 3-phosphate. Now, one most important thing is this glycerol which was going to the liver, which we talked about. Why is this glycerol not entering into the adipose cells and binding to the fatty acids? See, the triglyceride is broken down into fatty acid and glycerol. Fatty acid comes inside, but glycerol does not, and then glycerol is formed by the intermediate of the glycolytic pathway for the formation of triglycerides. Why is that happening? Is because glycerol 6-phosphate cannot cross the membrane first. So it has to be glycerol which crosses the membrane, but glycerol, if it crosses the membrane, because it is non-polar, glycerol cannot be converted into glycerol 6-phosphate because the adipose tissue does not have glycerol kinase. Because the adipose tissue does not have glycerol kinase, it cannot make glycerol 6-phosphate, and for the esterification of fatty acids of glycerol, it is required to first phosphorylate the glycerol and then it can esterify. This glycerol is useless for the adipose tissue. This glycerol is sent to the liver so that the liver can use this glycerol to form glycerol 3-phosphate by glycerol kinase, which the liver has, and esterify. Understood? So what is happening in the fed state is, in the liver there is increased glycolysis for energy, and there is glycogenesis, which is formation of glycogen or the storage of glycogen. Also there is lipid formation. In muscle tissue, what is muscle cells is happening is there is increased protein synthesis. Also there is glycogenesis plus glycolysis is also happening for energy. And in the adipose tissue there is lipogenesis. In the fasting state also we'll be talking about 3 organs. One is the liver, the second is the muscle, and third is the adipose tissue. Now one thing you should notice here is, in the previous slide, in the fed state, there was no interaction, there was little interaction between the organs because they were all happy and blood was rich with glucose, chylomicron and amino acids. They didn't rely on each other. But when the bad times come, when the fasting state is there, the intestine is not providing any nutrients, then they rely on each other for all the nutrients. So just like in life, now in the bad state, the liver, what will liver do? Liver will break down the glycogen. The glycogen will break down and form glucose. The glucose will go into the plasma via the GLUT2 transporter, because it is two-way transporter. Bidirectional transport can happen. The glucose which is in the plasma is given to the muscle so that the muscle can use glucose for the energy. Second is, the glucose is also used for some formation of some amount of pyruvate, and that pyruvate goes to acetyl coenzyme to provide energy. But the major energy which is coming from is from the fatty acids, which is provided by the adipose tissue. So this fatty acids will go into acetyl coenzyme A, beta oxidation, then go into the TCA cycle to provide energy. Also there is a cycle which is going, that the from muscle there is alanine coming, alanine goes to pyruvate, pyruvate forms glucose via gluconeogenesis, that glucose goes to the muscle. Okay? The glycerol is also coming from the adipose tissue, and that glycerol will also come and form glycerol 3-phosphate. This glycerol 3-phosphate can also form glucose via gluconeogenic pathway. In the muscle cells, what happens is there is a very small amount of insulin independent glucose uptake. So in fed state there was a high amount of uptake which was insulin dependent. In fasting state there is a slow amount of insulin independent uptake of glucose, and in exercise we'll see that there is a high amount of glucose uptake which is insulin independent. Okay? These three things you should remember. In muscle also, what happens is, muscle is a selfish organ, it will not give its glycogen to form the glucose. So what happens is, glycogen is broken down into glucose 6-phosphate, and that glucose goes to glycolysis, pyruvate, acetyl coenzyme A, and TCA cycle. But it will not contribute into plasma glucose. Why? Because muscle does not have glucose 6-phosphatase. So muscle cannot break down the glucose into glucose 6-phosphate into glucose, and the GLUT transporters are bidirectional transport for only glucose. They don't transport glucose 6-phosphate. So that glucose 6-phosphate which is formed by glycogen breakdown cannot go out. It can only go into the glycolytic pathway, and it forms energy. Second is, the pyruvate can go into lactate. Lactate can go to liver, form pyruvate, and then gluconeogenesis. So it is indirectly contributing to glucose. The alanine goes to liver, and then it also forms a part of gluconeogenic pathway. So indirectly the muscle is contributing to blood glucose levels. And also the fatty acids which was stored in the muscle is used for energy, or the fatty acids coming from the adipose tissue. Now in adipose tissue what happens? I already told you that in adipose tissue it is very simple. The fatty acids It is only concerned about fatty acids. So there is no uptake of glucose now because there is no availability of glucose. Most of the energy is coming from the triglycerides breaking down into fatty acids and glycerol. Glycerol is sent to the liver to form glucose via the gluconeogenic pathway. The fatty acid undergoes beta oxidation, forms acetyl coenzyme A, enters into the TCA cycle to form energy. Simple. So what is happening in the fasting state? In liver, now the gluconeogenic and glycogenolysis is happening. In muscle, the proteolysis is happening and glycogenolysis is happening. And in the adipose tissue, there is lipolysis happening. So this is how the metabolism in fasting state happens. Now in starvation, what is different? Like starvation is nothing but prolonged starvation. Prolonged starvation is more than 72 hours, but starvation is more than 24 hours of not taking a meal. What happens in this is in the liver, now the glycogen stores are also completely depleted. The glycogen stores are also completely depleted. So the liver is completely dependent on the fatty acids for energy. One. In starvation, what happens? Now the liver will start producing ketone bodies because the brain was like, now I will not just consume glucose, I want to spare the protein. Why spare the proteins? Because gluconeogenesis, the major substrate for gluconeogenesis is protein. So even if now the gluconeogenesis is taking place in the absence of glycogenolysis, there will be a large breakdown of proteins to sustain the metabolism of brain. Now the brain is like, I'll consume ketone bodies also. So liver will produce keto bodies from acetyl coenzyme A, but liver cannot consume those ketone bodies, because it does not have the enzyme thiophorase to consume the ketone bodies. That is a very crude level of compartmentalization. Like the liver is producing ketone bodies and the rest of the cells are consuming ketone bodies, because if the liver would be consuming ketone bodies, then there would be a futile cycle which would be happening. Like the liver produces ketone bodies and consumes. So that is a futile cycle and we don't want that. So liver does not consume ketone bodies, it sends the ketone bodies into plasma for other cells to use. Simple. Now the glycerol is coming from the fatty tissues like the adipose tissue, and the glycerol 3-phosphate will form the gluconeogenesis. Now there is in the muscle cells also, muscle will consume ketone bodies, muscle will consume amino acids, muscle will consume fatty acids. All of this muscle will consume because now the glycogen is depleted completely to produce energy. In the adipose tissue, nothing is happening. It just breaks down the triglycerides into fatty acids, and the fatty acids are used for the energy production. The glycerol is sent to the liver for gluconeogenesis. So this is what is the most difficult part of this lecture: the metabolism in fed state, fasting state, and starvation. All these pathways might seem very difficult, but if you revise this once, this would be very easy. So now I would recommend you to pause the video, take a 5-minute break, and then come back to it. Okay. So now we'll be going into the hormonal control of metabolism per se. Now we have talked about all the pathways which are happening in the metabolism in the fed, fasting, and starvation. Now how is this controlled by the hormone? So what would be the goals of hormonal control first? So goals is first important thing is to ensure the availability of energy substrate to all the cells, and second is to maintain blood glucose levels. So these are two different things. See, the energy substrate does not necessarily come from the blood glucose level, but we have to maintain both of these. The energy should also be provided to all the cells, and blood glucose should also be provided for glucose dependent cells. And the third is to conserve energy. If there is no hormonal control, then there might be futile cycles which will be happening. We'll be seeing it when the first slide comes back. We'll be seeing how the futile cycles are prevented. Then the mechanisms of hormone control would be feed forward control and feedback control. Feedback control is very simple. If the glucose is increasing, then there will be increased insulin. The insulin will decrease the glucose levels. That is the feedback control. But feed forward control is the incretin mechanism that you might have read in the GI lectures, that feed forward control is anticipatory control. Even before the glucose reaches the beta cells of the pancreas, and the beta cells of the pancreas can sense the glucose, there is release of incretin hormones which are like GLP or GIP from the L cells of the K cells of the intestine or the ileum to be specific, and these incretin hormones will act on the beta cells to release insulin. That is a feed forward control of hormone. Okay. So what is the hormone control? Mainly the insulin is acting opposite to all the other hormones. So glucagon, adrenaline, cortisol and growth hormone are known as counter-regulatory hormones because they counter the effect of insulin. Insulin will try to decrease the level of glucose, and all the other hormones will try to increase the level of glucose in the body whenever they are increasing or decreasing. So major action of the hormones are in insulin. What does insulin do? Insulin is an anabolic hormone per se, and glucagon, catecholamines and cortisol will increase the blood glucose to maintain the blood glucose, for the maintenance of blood glucose. Insulin will decrease the blood glucose, glucagon, catecholamines and cortisol will increase the blood glucose. And what will growth hormone do? Growth hormone will act with respect to protein metabolism. It will act as an anabolic hormone and it will act together with insulin. But with respect to carbohydrate or glucose metabolism, it will act along with all the counter-regulatory hormones for the maintenance of blood glucose. So see, there is nothing black and white in the medical science. So there can be the growth hormone does not necessarily have to be a catabolic hormone because it maintains blood glucose by increasing the level of glucose. It acts along with insulin when there is protein metabolism taking place, and in carbohydrate metabolism it acts along with all the other counter-regulatory hormones. Understood? Next, the Isle of Langerhans. So what is the Isle of Langerhans? Isle of Langerhans is the endocrine group of cells which are present in the pancreas. So there are two groups of cells. There is exocrine acinine and then there is endocrine isles. Endocrine isles take up about 4 to 5% or even less than that of the total pancreatic volume, and the endocrine isles are known as the Isle of Langerhans, and there are multiple cells which are arranged in the Isle of Langerhans in this fashion. See, these IHC stained molecules, the IHC stained cells are beta cells and these are the alpha cells. So this anatomy is very important for the control. See, beta cells are present in the center and the alpha cells are present in the periphery. The beta cells of the pancreas take up about 70 to 75% of the total volume, and the alpha cells take about 10 to 20%. The beta cell will secrete insulin and the alpha cells will secrete glucagon. So why is this anatomy important? Is because of the blood flow to the Isles of Langerhans. The blood flow to the Isle of Langerhans is centrifugal. So the blood arterioles which project into the Isle of Langerhans will take the blood into the center. So the first cells which encounter the blood coming from the portal circulation is the beta cells of the pancreas. Now the beta cells of pancreas, if the blood had a large amount of glucose, beta cells of the pancreas will release insulin. Now this insulin will be secreted into the blood. Now this blood vessels or the blood containing insulin will go outwards and act on the alpha cells, and they will inhibit the alpha cells to release glucagon. The glucagon is released in a basal tone. It is released every time until and unless it is inhibited by the insulin. So if the blood containing glucose comes to the beta cells, it will lead to release of insulin. That increased insulin rich blood would go outside and inhibit the alpha cells to release glucagon. So that is how the blood flow or the anatomy of the beta cells and the alpha cells is very important. Understood? Now the regulation of endocrine pancreas. How does the regulation of endocrine pancreas takes place? It can take place via the vascular route, the neural or the local route. So from the blood vessels there are hormones which will inhibit or activate the beta cells to release insulin, or the nutrients. The most important regulator of insulin release by the beta cells of pancreas is glucose, which is the nutrients. Then there is nerve impulses or the neuropeptides which are acting on the beta cells. There are paracrine influences from the other cells, or there are gap junctions which act via the electrical coupling. So we'll be talking about the regulators of insulin secretion. So the primary stimulus for insulin secretion is blood glucose levels. So glucose in the blood enter via GLUT2, which is insulin independent, into the cell. As soon as it enters into the cell, the glucose is converted into glucose 6-phosphate via glucokinase. Glucokinase is present in two cells of the body, mainly, which is the beta cells of the pancreas and the liver cells. Yeah, so the hepatocytes. So the glucose goes to glucose 6-phosphate via the glucokinase, and the glucokinase has a similar Km as the physiological range of the glucose, which is around 70 to 150 mg per deciliter. So at this level of there is regulation of glucose. So what is the difference between regulation and control? In regulation, there must be a sensor. There must be a sensor which senses the level of glucose in the body. So in the beta cells of the pancreas, that sensor is the glucokinase, because the Km of the glucokinase is similar to the physiological range. So whenever the glucose comes inside, the glucose will be converted to glucose 6-phosphate. Now the glucose cannot go outside because it is trapped in glucose 6-phosphate. It enters the glycolytic pathway and forms ATP via the TCA cycle, and that ATP now will block the ATP regulated potassium channels. Will block the ATP regulated potassium channels. Now the potassium cannot go outside. Now there will be more potassium. More potassium leads to depolarization of the cell membrane. The depolarized cell membrane will activate the voltage gated calcium channels. The voltage gated calcium channels will lead to exocytosis of the secretory vesicles in which insulin was already stored. Now the exocytosis will lead to increased insulin and C-peptide in the blood. That is how release of insulin takes place. Understood? Now we also talked about the feed forward mechanism of incretins. So this is how the incretin acts. The glucose in the intestinal lumen, anticipatory and pervasive stimulus via the intestinal L cells. It will be sensed by the intestinal L cell. Intestinal L cells will release GLP-1. GLP-1 will act on the GLP-1 receptors, which is a G protein coupled receptors of the GS subtype. This will activate the adenylyl cyclase, leading to increased cyclic AMP, and the cyclic AMP will lead to increase in the protein kinase A. Protein kinase A will lead to exocytosis. So this is how the anticipatory or the incretin feed forward mechanism takes place. Now how can we use this to treat patients of diabetes? So in GLP-1 is degraded, or the enzymatic degradation takes place by DPP-4 enzyme. So if we inhibit the DPP-4 enzymes, then there will be increased levels of endogenous GLP levels. So these DPP-4 inhibitors have been made. For example, sitagliptin or other gliptins are the DPP-4 inhibitors which are used for the treatment of diabetes. DPP-4 resistant GLP-1 analogs, if I give, for example, liraglutide is a drug which is a GLP-1 analog. It will directly act on the GLP-1 receptor and it will lead to insulin secretion. Another kind of drugs which are the sulfonylurea based oral hypoglycemics, which are the glibenclamide or glipizides, sulfonylurea based drugs. They will directly block the ATP regulated potassium channels. So if they block, then all the steps forward will be taking place and the insulin will be released. So all these three groups of drugs, which are DPP-4 resistant GLP-1 analogs, DPP-4 inhibitors and the sulfonylurea based oral hypoglycemics, are known as insulin secretagogues. They will increase the secretion of insulin from the beta cells of the pancreas, but later in the course of the disease, they might lead to beta cell exhaustion. That you have to consider. So that was the primary stimulus, the glucose. The secondary stimulus for the insulin secretion is the parasympathetic innervation. ACH is acetylcholine. Acetylcholine acts on the M3 receptor, which is a GQ type of receptor, which will lead to increase in calcium inside the cell via the IP3DAG pathway, and that increased calcium will lead to insulin release. There is amino acids also, which are via the amino acid transporter goes inside the cell and will lead to, maybe because of the oxidation of amino acid leads to increased ATP, and the same mechanism of insulin release takes place, but we don't necessarily know how amino acids will lead to increase in insulin secretion. But there is also sympathetic stimulation, in which alpha 2 is the major sympathetic stimulation, And when the alpha 2 is stimulated via the norepinephrine or epinephrine from the sympathetic innervation, the cyclic AMP will decrease because it is a G1 type of G protein coupled receptor. So decreased cyclic AMP will lead to decreased insulin secretions. This is an inhibitor. The sympathetic system will inhibit the release of insulin, rest all will increase the release of insulin. Understood? So now insulin and glucagon, what happens is when we take a meal, the level of glucose, this is on the x-axis is the time after taking the meal, and on the y-axis is the levels of glucose in the first graph, insulin and glucagon in the second and third graph. So whenever we take a meal, there will be increased glucose. That increased glucose will lead to an increased insulin secretion via the mechanism which we saw, and that insulin, when it goes outside, it will inhibit the alpha cells to decrease glucagon. Glucagon was already being secreted. Now the glucagon secretion decreases. So in fed and fasting state, what happens in fed state, the insulin is dominant hormone. It will increase the glucose oxidation. Whatever pathways were happening in the fed state will be activated by insulin, and whatever pathways were happening in the fasting state will be activated by glucagon. As simple as that. So if you remember most of the pathways which were happening, then you don't have to cram anything here. Just have to remember whatever was happening in the fed state, like glycolysis, glycogenesis, lipogenesis and protein synthesis will be activated by insulin. And another trick is insulin will always dephosphorylate. So whatever enzymes are required for these pathways will be activated in their dephosphorylated state. Insulin will dephosphorylate the enzyme will be activated, and then these pathways will happen. Completely opposite will happen with glucagon. Whatever pathways were activated in the fasting state will be activated. The enzymes which activate those pathways will be activated in the phosphorylated state, and that phosphorylated state, the glucagon will phosphorylate that enzyme. So regulation of glucagon, how does it happen? This is just a summary of beta cells of the pancreas. Like epinephrine inhibits, sympathetic system will inhibit the insulin secretion. Glucose will stimulate the insulin secretion. Amino acid, fatty acid will stimulate. What you have to see here is, again, the medical science is not black and white. Amino acid will stimulate insulin, but amino acid will also stimulate the release of glucagon. The rest, everything is opposite. Like glucose will stimulate insulin, but glucose and insulin will inhibit glucagon. Sympathetic system, which is the epinephrine, is a very important regulator of glucagon secretion, and it will activate the alpha cells to release glucagon. So amino acids will stimulate both insulin and glucagon. Why does that happen? Is so as to prevent hypoglycemia after taking a protein rich diet. So what happens is amino acid will stimulate insulin after taking a protein rich diet. If the amino acid didn't stimulate glucagon, what would have happened is the X amount of insulin which was released, but the absorption was only Y amount of glucose, and the X amount of insulin will lead to decrease in the blood glucose level because it is more than enough for the Y amount of glucose. You understand? The amino acids led to the stimulation of insulin, and there was now an excess of insulin which was released in the blood, which was not required. Now this insulin will lead to decrease in blood glucose level below the level which was required. That is why the amino acid also stimulates glucagon, so that glucagon will lead to increase in blood glucose level and prevent hypoglycemia. This is the integrated regulation of the blood glucose levels. The three important hormones are the insulin, glucagon and the catecholamines, which is the epinephrine and the norepinephrine. If the blood glucose level is increased, there will be stimulation of beta cells of the pancreas. Insulin will be released, which will lead to decrease in the blood glucose levels. This increased insulin will inhibit the alpha cells of the pancreas. Alpha cell will decrease release of glucagon. Now, if the blood glucose level go below the normal limits, then decreased levels will now stimulate the hypothalamic centers and release the catecholamines. Also, the low blood glucose level will inhibit via the catecholamines will inhibit the beta cells of the pancreas. Now the inhibition of the insulin on the alpha cell is not there. So alpha cells can release glucagon, and the catecholamines also stimulate the alpha cells to release glucagon. So when there is low blood glucose, glucagon and catecholamine are increased, which lead to increase in blood glucose and bringing it back to normal. Now the actions of cortisol. Cortisol in the interdigestive period or the fasting state. There is stress during the interdigestive period. There is increased cortisol. There is increased epinephrine, norepinephrine, and there is increased glucagon, which means there is decreased insulin glucagon ratio. The liver, there is increased glycogenolysis and gluconeogenesis, whatever is happening in the fasting state. Skeletal muscle, the most important thing which cortisol does is in the skeletal muscle, it will lead to increased proteolysis. It will mobilize the proteins from the skeletal muscle to the liver so as to form the substrates for the gluconeogenic pathway. That is the most important thing what cortisol does. Rest everything is same. Protein synthesis will decrease, there will be increased glycogenolysis, there will be decreased uptake of glucose, in adipose tissue lipolysis will increase, and everything else is the same. Most important thing is mobilization of protein for gluconeogenesis. Action of growth hormone we talked about already is in protein synthesis. When protein synthesis is happening, protein metabolism, growth hormone acts along with insulin. When there is increased protein intake, increased growth hormone, increased insulin, increased protein synthesis. So it acts as an anabolic hormone. But when the carbohydrate intake is increased, the insulin is more, growth hormone is less. So caloric storage is increased. But when there is fasting state, growth hormone will increase along with the counter-regulatory hormones to increase the caloric mobilization. Understood? This is what was there about the other hormone, the cortisol and the growth hormone, and we talked about the insulin, glucagon and catecholamines in quite a lot of details. Okay, so now coming back to this dreaded slide, so we'll take it one by one. So first, glycolysis. So what happens in glycolysis is glucose is phosphorylated to glucose 6-phosphate by the enzyme hexokinase. Glucose goes to glucose 6-phosphate. I have only marked the irreversible steps. All the reversible steps, whichever enzymes are taking place, does not matter. By the physiological point of view, you can read about the details of each pathway in biochemistry. From physiological point of view, we'll only be talking about all the irreversible steps only. So glucose going to glucose 6-phosphate, then converted to fructose 6-phosphate, then fructose 6-phosphate going to fructose 1,6-bisphosphate by the enzyme phosphofructokinase 1, goes to fructose 1,6-bisphosphate. Then it is broken down into 3 carbon compounds, glyceraldehyde 3-phosphate, which we talked about lot of times, glycerol 3-phosphate. This is the same thing, glyceraldehyde 3-phosphate, which was taken up from the intermediate of the glycolytic pathway and esterified to fatty acids for triglyceride formation and DHA. Then it forms phosphoenol pyruvate. Phosphoenol pyruvate goes to pyruvate by the enzyme pyruvate kinase. Now phosphoenol pyruvate goes to pyruvate. Pyruvate goes to acetyl coenzyme A by the enzyme complex pyruvate dehydrogenase. It is not just a single enzyme, it is an enzyme complex which is pyruvate dehydrogenase, and then the acetyl coenzyme A, which is formed, binds to oxaloacetate to form citrate, which goes into the tricyclic acid cycle or the citrate cycle, Krebs cycle or TCA cycle. Then they form ATP or NADH, which the NADH which is formed goes to the ETC cycle, which is the electron transport chain. Now, how does the glucose is formed via the gluconeogenic pathway? Now, these all irreversible steps, we have to just find an enzyme which would reverse these steps, and the rest all pathways will happen automatically. The first step, which is the PEP to the pyruvate. Now we have to form PEP from pyruvate. It happens via a 2-step pathway. So pyruvate goes to oxaloacetate via the enzyme pyruvate carboxylase. Oxaloacetate is converted into malic acid, and then malic acid is sent to the cytoplasm, and that malic acid is again converted to oxaloacetate, and then the oxaloacetate goes to PEP via the PEP carboxykinase. That PEP goes via the reversible steps to fructose 1,6-bisphosphate, and then it is by 1,6-bisphosphatase goes to fructose 6-phosphate, then a reversible step, then the glucose 6-phosphate is converted to glucose via the glucose 6-phosphatase. Simple. So all this is how the enzyme regulation is taking place, and now you can guess which enzyme would be activated by which hormone and which enzyme will be deactivated by which hormone, which will be activated in their phosphorylated state and which will be activated in their dephosphorylated state. It is that simple. Now glucose 6-phosphate, not glucose, this is a mistake. The glucose 6-phosphate goes to glucose 1-phosphate, and then glucose 1-phosphate goes to glycogen via the ninth enzyme, which is the glycogen synthase. So glucose 1-phosphate going to glycogen synthesis, then glycogen going back to glucose 1-phosphate via the glycogen phosphorylase, and the glucose 1-phosphate will go to glucose 6-phosphate. So now see, all the pathways are now making sense. Now why did the muscle not muscle not able to contribute to blood glucose? Because the glycogen goes to glycogen 1-phosphate to glycogen 6-phosphate. Now I need this enzyme glycogen 6-phosphatase to convert it back to glucose, but the muscle did not have that enzyme, so the glucose 6-phosphate goes directly to pyruvate and is providing energy. In fructose metabolism, we'll be talking a lot in detail in applied aspects. Lipid metabolism is very important. Acetyl coenzyme A, excess acetyl coenzyme A goes via acetyl coenzyme A carboxylase to malonyl coenzyme A, which forms fatty acids. Fatty acids, which we have talked multiple times, binds to esterified to glycerol 6-phosphate to form triglyceride. This is the fatty acid synthesis or triglyceride synthesis. Now the breakdown which happens in the fasting state is triglycerides are broken down into fatty acid and glycerol. Glycerol goes into the intermediate to form gluconeogenesis, to form glucose, and the fatty acids via the beta oxidation forms acetyl coenzyme A. That acetyl coenzyme A enters the TCA cycle to form energy. Simple. And in the starvation state, what happened was the acetyl coenzyme A, now a lot of acetyl coenzyme A binds to each other and form acetyl coenzyme A, which is broken down into acetoacetate and beta hydroxybutyrate, which are the ketone bodies, in which the beta hydroxybutyrate is the main ketone body. So ketone body synthesis and breakdown, both of them are catabolic pathways because they are formed by breakdown of something. In protein metabolism, nothing happens, but amino acids will be deaminated to form intermediates of the TCA cycle. They enter the TCA cycle and provide energy. So now you understand, like, all the metabolisms are interrelated to each other. Nothing happens isolated in the body. All of them are connected to each other, and so if one of the pathways is affected, then all the pathways will be necessarily affected. Understood? So now, guess insulin, what will insulin activate? Insulin will activate glycolytic pathway, so it will activate the hexokinase, all the enzymes which are required for glycolysis, and it will inhibit the opposite pathway. Why? So as to prevent futile cycles. So all these enzymes 7, 8, 5 and 6 were activated in their phosphorylated state. So when insulin dephosphorylates them, they will be inactivated, and all these enzymes 1 to 4 will be activated in their dephosphorylated state. So insulin will be activating them. Insulin will do nothing. It will just dephosphorylate all the enzymes. Some enzymes will be activated, some enzyme will be inhibited, and the body does it beautifully, that the enzymes which are glycolytic enzymes will be activated and gluconeogenic will be inactivated. Glycogen pathway, what will happen is glycogen synthesis enzyme will be activated and the glycogen phosphorylate will be inhibited. In lipid metabolism, the formation of lipid will be activated and the oxidation of lipid will be inhibited. Simple. Doing once again, glucagon, what will glucagon do? It will activate the gluconeogenic pathway, inhibit glycolysis because it will phosphorylate. Second, it will inhibit the glycogen synthesis, it will activate glycogen breakdown, and it will activate the beta oxidation and inhibit the fatty acid synthesis, triglyceride synthesis. Simple. So now all of this is very simple now. So now we'll just a couple of more slides about the applied aspects is left. Metabolism during exercise. So in this, there x-axis there is time and y-axis there is the source of energy. So first few seconds of energy would be provided by the stored ATP and creatine phosphate, but that is a very little amount of energy. It will not sustain more than a few seconds. So for the next few minutes, what will happen is the anaerobic glycolysis will take place because that is fast. So muscle glycogen would be broken down, and the glucose which is present in the blood will be broken down via the anaerobic glycolysis, and the ATP or the energy will be provided by that. But that does not happen more than few seconds. Why? That is not just because the glycogen is being depleted, because glycogen stores are there for few hours. That is because via the anaerobic glycolysis, lactic acid has been accumulating, and that lactic acid will inhibit the anaerobic glycolysis. Now the aerobic oxidation will take place. 66% of the aerobic oxidation would take place via the fatty acid oxidation, which comes from the plasma or the adipose tissues, and the rest 33% would be carbohydrate metabolism, would be muscle glycogen, plasma glucose, or the liver glycogen. So during exercising muscle, what is there is decreased insulin and increased glucagon, which is important, but what is much more important is the increased sympathetic load. During fasting state, the glucagon insulin ratio was similar to this, but the catecholamines were relatively lesser. Catecholamines increased, but they were relatively lesser to this. And the most important thing is insulin independent glucose uptake, which is very high. So during fed state, the insulin dependent glucose uptake is high. During fasting state, insulin independent glucose uptake is low. And during exercise, the insulin independent glucose uptake is high. Understood? The second most important thing would be the endocrine response to hypoglycemia. Not talking about diabetes in this lecture, we talk about hypoglycemia. So in hypoglycemia, what happens is whenever there is blood glucose less than 50 mg per deciliter, the hypothalamic regulatory centers are activated. One. Second, the alpha cells of the pancreas will be activated and they will release glucagon. The increased glucagon will what will it do? It will increase the glycogenolysis and it will increase the glyconeogenic path. Simple. Second, the low blood glucose will inhibit insulin. It will activate the hypothalamic regulatory centers, which will stimulate the autonomic nervous system. And now the sympathetic nervous system is activated. The sympathetic nervous system, the epinephrine and the norepinephrine will inhibit insulin, and it will activate the alpha cells of the pancreas to release glucagon. They will act directly to increase the glycogenolysis, the epinephrine and the norepinephrine, and via the hypothalamic pituitary axis, adrenal axis, they will increase the ACTH, which is the adrenocorticotropic hormone, which will lead to increased cortisol. And we saw the role of cortisol in increasing the gluconeogenesis via the mobilization of protein. So all these hormones will be increased and they will counter the low blood glucose level. So what is the time dynamics or the level of blood glucose at which all of this is happening is below 85 something, the insulin production decreases. Then 60-65, the epinephrine and glucagon production, because these were the two hormones which were very important for increasing glucose. So these will increase first. Then there will be growth hormone increase. Then cortisol levels will increase for mobilization to increase the gluconeogenesis. Now below 55, the adrenergic symptoms starts increasing. Now the catecholamine, the autonomic nervous system, which is the sympathetic nervous system which is activated, will not only increase the glucose level, but it will also lead to anxiety, palpitation, tremor and sweat. Second, below less 50 mg per deciliter, neuroglycopenic symptoms will happen. These neuroglycopenic symptoms would be headache, confusion, slurred speech, and eventually they might lead to seizures, coma, and even death. Understood? So now why is beta blocker contraindicated in diabetic patients? You can understand this. Why is beta blocker contraindicated? Is because first, the beta blockers will block the effect of all these hormones on the liver. So now norepinephrine, epinephrine cannot act on the liver and cannot lead to glycogenolysis. They cannot increase gluconeogenesis. So now the body's counter-regulatory response to hypoglycemia is inhibited. First. Second, all these symptoms will also be blocked by the beta blockers. So there will be no anxiety, palpitation, tremor or sweating. So now the patient is going into hypoglycemia. The body cannot counter the hypoglycemia. And third, the patient does not even know that he or she is going into hypoglycemia. So that is why beta blockers are contraindicated in hypo. Understood? Now we'll come to the third applied aspect, which is the fruit juices, which are bad for our body. Why? Because it has a high amount of added sugar, plus it has highly concentrated corn syrup, which is nothing but fructose. And via our pathway, we know that fructose goes to fructose 1-phosphate, that fructose 1-phosphate goes to glyceraldehyde 3-phosphate, and it enters directly into the glyceraldehyde 3-phosphate pathway. It bypasses all the rate limiting enzyme. So now bypassing the rate, what is the rate limiting enzyme? It slows down the process. Now if the fructose bypasses the rate limiting enzyme, it will go into the pyruvate and acetyl coenzyme very fast. Now the acetyl coenzyme A is much more acetyl coenzyme is being produced than the capacity of TCA to use it. And now the excess acetyl coenzyme A will go into fatty acid synthesis and the triglycerides will be formed. Fructose is the most lipogenic substance, one of the most lipogenic substance, because it will directly go to acetyl coenzyme A and then form triglycerides and stored as lipid in the body. One. Second, these fruit juices are liquid, and the liquid don't have a very long gastric emptying time. So there will be a faster gastric emptying time, go into the small intestine. Second. Third, they don't have any fibers. The dietary fibers are negligible in the fruit juices. So the absorption of the glucose will be very high, very rapid and very high. The peak of the glucose in the blood after the consumption of these fruit juices will be there. So all these three reasons are why the fruit juices are very lipogenic, and they will lead to increased lipid synthesis in our body. Understood? So to summarize, whatever we have understood is, in the absorptive state which we were talking about, that is the fed state, and the fasting state or the post-absorptive state. What happens is, the absorptive state, there is increased nutrients in the body, in the intestine. Fasting state, there are no nutrients. So these nutrients will go via the portal system into the pancreas, and in the pancreas they will lead to increased release of insulin by the beta cells. Fasting state, there are no nutrients, so insulin is not released, so glucagon is increased in the alpha cells. In adipose tissue, what happens is, so now in the three organs, and the fourth organ is brain. So in the adipose tissue, nothing but increased synthesis of triglycerides. In here, the increased utilization of triglycerides, fatty acids. In liver, there will be glycolysis, glycogenolysis. No, sorry, not glycogenolysis, glycogen synthesis, triglyceride synthesis. In fasting state, what happens is, there is glucose produced by, this is gluconeogenesis and glycogenolysis. And also in starvation, there is ketone body formation. In muscle fed state, glycogen will be formed, protein will be formed, and in fasting state, the fatty acids and the ketone bodies will be used by the body. And the brain will use glucose completely oxidized to carbon dioxide water in fed state, and in fasting state also it will use glucose, but in the later stages of starvation and prolonged fasting, it will start to utilize ketone bodies. So that is what is happening, and it is not it appears to be a very overwhelming thing. It appears like that there is a lot to cram about, but most of the things are just the repetitions. If you understand the pathways, the first half of the lecture, if you understand that, then the rest of the lecture is very easy to understand, and how insulin glucagon and that. This will also help you in diabetes and the endocrine lectures of insulin and glucagon in the beta cells of the pancreas, the alpha cells of the pancreas. Okay, and so if you have any doubts about this lecture, you can either comment on this video, or you can WhatsApp or mail, or you can DM on the Instagram handle. You can reach out to us, and we'll try to get back to you as soon as possible. Okay, thank you, and we'll try to keep posting more videos regularly. Thank you.